By far the most important question of the pandemic is, what is the origin of SARS-CoV-2? This diagram reveals the origins of two close relatives of SARS-CoV-2, members of the beta coronavirus genus, MERS-CoV and SARS-CoV-1. In both cases, the original virus came from a bat, which infected camels. In the case of MERS-CoV, or Middle East Respiratory Syndrome, CoV, and then on to the final host, humans, through close contact with mucus or fluids. Not surprising, individuals in this part of the world ride camels, they eat them for meat, and so there's lots of close interaction between these species. In the case of SARS-CoV-1, it seemed pretty clear that it was a bat virus that jumped into a civet cat, changed enough to then jump into the final host, humans. In the parts of the world where this was discovered, individuals eat civet cats, and they also use them in some production of a weird civet cat coffee. So there is lots of uses for civet cats, and obviously lots of opportunity for direct or close contact. However, in the case of SARS-CoV-2, there was lots of hypotheses as to where it came from. Clearly, it came from a bat originally, but what was in the middle here? At first, it was thought pangolin, because pangolin and SARS-CoV-2 viruses have very similar regions of their spike protein. But when you look at the overall similarity of the RNA genomes of the viruses, they're very different. There's absolutely no way that the virus recently jumped from pangolins to people. The viruses are just simply too different. So the question is, what's here? What happened here between bat and human? So is it some unknown animal that we've yet to discover? Very unlikely, given the fact that we've sampled hundreds of thousands of RNA sequences from coronaviruses and tens of thousands of animals and never found anything like a beta version of SARS-CoV-2. The only thing that's been studied right now is the human-adapted SARS-CoV-2. There's no intermediates that have yet to be found. In the case of SARS-CoV-1, the intermediate was found within months of the pandemic starting, and that was back in 2003, when arguably DNA sequencing technology was in its infancy. Here we have a variety of techniques for rapid, high throughput analysis, and we still have yet to find anything like a beta version of SARS-CoV-2 in nature. That led certain researchers like Nikolai Petrovsky at the University of Flinders to suggest that it may have come from evolution in a laboratory, in which viruses from bats were grown in humanized cell culture to basically become optimized for human infection. And so you can watch this video if you're interested in that concept, but I'll talk about some other potentials as well. Not just cell culture, but deliberate genetic engineering in order to create a highly transmissible SARS-CoV-2 virus. So the Petrovsky work that was published in, I believe, Nature, is titled In Silico Comparison of SARS-CoV-2 Spike Protein, ACE2 binding affinities across species and implications for virus origin. So Nikolai's lab basically took the amino acid sequences of the earliest SARS-CoV-2 spike proteins and compared them to the amino acid sequences of ACE2 from a variety of animals that were believed to be the origin species for SARS-CoV-2, and basically used these computer modeling software to figure out which animals have the highest binding affinity. And surprisingly, they discovered that the spike protein exhibited highest binding to human ACE2 of all the species tested, which sort of defies evolutionary logic. If a virus recently jumped from a pangolin, for example, then it should have the highest binding affinity to pangolin ACE2. Again, that was not the case. Another bit of intrigue here is that this paper was submitted for publication in, I believe, summer of 2020, and it was not accepted for publication until the following summer of 2021, in line with when Dr. Fauci had finally admitted that a lab leak was possible. And again, that's highly suspicious, and it strongly suggests censorship at the highest level of scientific journals. So when we look at closer detail of the data, we find a few anomalies in the genome. So here's a layout of the genome showing you the RNA sequence basically broken into regions. Open reading frame 1 and 1b. This region largely encodes replication enzymes. So for example, the polymerase that the virus needs, and the proteases are in here somewhere that are required to process the polyprotein. Later in the genome, you start to see the structural proteins. Here's the spike region. This is the region of the RNA that encodes the spike protein. I should point out that this is a really big genome as far as viruses go, 27.9 kilobases. So this right here is just a stretch of RNA right here at this midpoint of the sequence. And this right here encodes what I referred to previously as the furin cleavage site. That would be this short stretch of RNA right here. The whole RNA sequence for the spike is about 3,821 nucleotides, so this is quite a short region. But the point that I wanted to make is that this furin cleavage site that occurs right here is not found in any of the closest siblings to SARS-CoV-2. And again, the sequence is very powerful. It gives the virus incredible affinity for human host cells. As I already mentioned, it also helps in the formation of syncytia, which allows the virus to optimally spread from cell to cell. So this region here also has another anomaly, and in that you see the codons. So this codon encodes a proline, this codon encodes arginine, this codon encodes arginine, this codon encodes alanine, and this codon also encodes arginine. So the more arginine-rich the furin cleavage site is, the more efficiently it is cleaved. So you can imagine that this proline, for example, would be acted on very swiftly by evolution as the virus spread through human population. And indeed, that's what happened. Once we got to Delta, which presumably was a better killer overall, the virus mutated this C right here to a G, which then made this codon CGU. And so from Delta on, the virus had an RRRAR furin cleavage site, became more efficient. So for example, Omicron also has that, and so became more efficient and spread much easier. But the other thing I wanted to point out too is that in this set of codons, it's very unusual that the virus would use CGG to encode arginine. There's actually 6 different codons that can be used to encode arginine, and of the 6, SARS viruses don't regularly use CGG. They, in fact, very rarely use CGG to encode arginine. And so the fact that there are 2 of these right next to each other, the only time that occurs in any of the SARS-like viruses is right here in this very important region. These 2 CGGs right next to each other is highly suspicious. And in fact, when I posed this to a colleague of mine, the person said, human gene jockeys commonly use CGG for arginine. Is that a complete coincidence? So basically, when somebody wants to encode an arginine-rich protein in an animal cell, a human cell, they'll use an RNA fragment with CGG in it. And so again, this is a signature for genetic engineering, having these CGGs right next to each other, in addition to the rarity of the furin cleavage site. So another way to look at this is through restriction mapping. And so when we want to genetically modify microbes, we tend to use restriction enzymes in order to cut the genomes at specific places in order to insert and remove pieces of RNA or DNA. And so BSA1 and BSMB1 are 2 examples of restriction enzymes that can be used for viruses. And these are naturally occurring, but you can also insert them in specific places in order to get desired outcomes. And so if you have a virus that's genuinely natural, that absolutely came from nature, no question about it, you expect a random distribution of these restriction enzyme sites. So all these little red balls are BSA1, and all the little bluish ones are BSMB1 sites in the genomes of these various microbes. Notice how, again, here's SARS-1, for example. Notice the sort of random distribution of these throughout the genome. But when we look at SARS-CoV-2, here's WIV04 and here's Wuhan 1. These are the two SARS-CoV-2 viruses. Notice the unnaturally consistent distribution of these throughout the genome of these viruses. Again, strongly suggesting, as these researchers concluded, this indicates SARS-CoV-2 likely originated from reverse genetic system. And again, looking back to the furin cleavage site, if you line SARS-CoV-2 and its region of the spike protein that I showed you previously with other close relatives, specifically in the beta coronavirus group, nobody has anything like the PRRAR in this particular region. The yellow regions I highlighted to show you places of 100% identity. So there's a fair amount of 100% identity throughout, but the fact that there's just absolutely nothing here, the computer actually had to put physical spaces in here to realign the rest of the sequence once we got to the furin cleavage site. And when you look at, this is a publication from Alina Chan at MIT. If you look at all these really close relatives of SARS-CoV-2, and here's that region again, a little bit more of that region, but you see the identities highlighted in different colors of different amino acids. Only Wuhan 1, only of all these, so only SARS-CoV-2 has the furin cleavage site, the PRRAR. Nobody else has that. And so it strongly suggests, and we have lots of papers from 2018 and prior, that researchers specifically talk about inserting furin cleavage sites to increase transmissibility. The goal in a lot of these cases was to preemptively develop vaccines for viruses that did not yet exist. And so they inserted these furin cleavage sites to assume where evolution would head and therefore make vaccines for these preemptively. So again, this presence of a furin cleavage site in SARS-CoV-2 and none of these other viruses, when you take it in the context of the prior work that was being done to design viruses with furin cleavage sites that didn't naturally have them, it starts to present itself as a suspicious story. So what is the origin of SARS-CoV-2? Is it an accidental lab leak of a CoV-2 strain that was optimized by passage in humanized mice or other cells? That seems to be very plausible. Again, by humanized mice, we're talking about cells or mice that express human ACE2. Was it an accidental lab leak of a deliberately genetically modified strain? That's very plausible as well, given the furin cleavage site, given the CGG codon anomalies and the restriction patterns. Was it a natural transmission from an animal? That's also possible, but highly unlikely, and there's absolutely zero evidence for that at this point. The SARS-1, for example, was believed to have emerged from the wet market, but at least in that case, we found a beta version of the virus fairly quickly. Here with SARS-CoV-2, the only thing that's been discovered is the highly human-adapted version. There is simply no example of SARS-CoV-2 in nature. A natural beta version of SARS-CoV-2 simply does not exist after millions, literally millions of RNA sequences have been analyzed. What other scenario could there be? And these are important questions as well. What are the ramifications of each scenario? And obviously the ramifications are not good. If you're the United States, which presumably funded a lot of this research that was happening in Wuhan, China, to create strains of virus with furin cleavage sites. What other data do we need to really solidify this? Well, the researchers that are in favor of a natural origin need to go out into the world and slog through jungles and sequence viruses from as many animals as possible. I don't know how many more though. I mean, like I said, we've already analyzed millions of sequences.